Hello everyone, welcome to my channel. So today we are going to discuss about biopolymers. So far in the last two classes what we discussed, biopolymers is almost we know now what is biopolymer. Half of the thing is done. So biopolymers are polymers produced from natural resources. Biopolymers are large molecules made up of monomeric units. It's a definition of the same polymers, but here it should be made. These are obtained from the living organism. This sentence should be there. Without this sentence, you are just defining polymers. Okay. So here the polymer definition only, but here these biopolymers are made from what, or obtained from what, or extracted from what, or isolated from living organisms. Any word you can use. So these can be either be chemically synthesized or from the biological materials, or by synthesized by the living organism itself. For example, there is some polymer like for example we are having cellulose from the plant, but I have extracted that by using some chemical techniques. Okay, then you can call it as chemically synthesized from biological materials. Understanding? Or if I am using a DNA from a particular this thing, or just a particular cells or tissues, then we can call it as a biosynthesized directly by living organisms. Understanding the difference? So if you are using any method to extract or isolate, that becomes chemically synthesized from the biological materials. Or else, if you are using directly, then you can call it as a biosynthesized by living organisms. So these are made up of the monomeric units bonded together by covalent bonds. These monomeric units form a large molecule, that is biopolymers. So as biopolymers are derived from the living organisms like plants and microbes, they are renewable resources that we just discussed in a previous class. And unlike most polymers, which are petroleum based polymers, same thing in the table we have discussed. So generally the biopolymers are degradable in nature. All the biopolymers are degradable in nature. They find use in various industries, ranging from the food industries to the manufacturing industry and packaging industry. Okay, and obviously the last application we are only studying this particular thing, that is biomedical engineering applications. Biopolymers are promising materials owing to their characteristics like abundance in nature. They are available in plants. Okay, the most abundant polymer on earth today is cellulose, that is from the plant, that is a biopolymer. So that's for example I'm giving. So next, they are having the best properties whatever we need to use in order to use them inside the human body. That is, biocompatibility is very good in natural biopolymers. Then unique properties like non-toxicity. Okay, whereas the synthetic polymers exhibits toxicity, whereas biopolymers will not exhibit any kind of toxicity. So with some nano-sized reinforcements, that means at the nanotechnology, so at nanoscale, that is less than 200 or 300 nanometer, we can reinforce or you can tune some of the properties with some nano-sized reinforcement to enhance its properties. Practical and as well as practical applications, biopolymers are being researched for its use in more and more possible ways. Now nanotechnology might be some new word for you guys. So let me tell you what is nanotechnology. Nanotechnology is not something new technology. Nanotechnology existed since 400 BC also, but they didn't know the technology. They knew how to do certain things. For example, they like this cup is one of the famous cup, okay, which is from the almost maybe 4th century or something like that. It is still preserved in the museum, British Museum. So that is famous for its optical properties. That means when the light is focused on this particular cup from outside, it will appear red in color, but green in color in the refracted light or in this thing. And when the light is transmitted through the cup, inside the cup, the whole glass will turn into red in color. Isn't that amazing? So the people, those who were doing in 4th century, they knew how to make it, but they didn't know the reason why it is happening like this. The reason we only came to know after the development of electron microscopes. Even though after optical microscope also scientists have went and scratched and searched everything, but they didn't find anything, because if any particle size falls less than the half of the de Broglie wavelength, that is around 325 nanometer, so we cannot see that in the optical microscope because light will cannot bend in order to view that particular such things in optical microscope. So we need to change this optical, that is light source, light beam with the we can replace this light beam with the electron beam, so that we can see even 0.1 nanometer also. Understanding? So once the electron microscope development happened, then the scientists again went and scratched this particular thing. Let's see if any lesser particles are there or not. And they were amazed to see the existence of the gold and silver colloidal nanoparticles inside wall of this particular, what do you say, the scrub. So here what I am trying to say is that, so at nanoscale all the properties will change, and nanoscale is nothing but in between the atomic and molecular scale. And what are all these polymers, all those things? They are molecules, right? And molecular size is around 10 to the power minus 8, whereas in nanoscale it is 10 to the power minus 9. That means we cannot just say 10 to the power minus 9. The range has to be 1 to 200 nanometer. So in that particular technology, whatever the science is there, that is totally different from the bulk. Okay, we know that atomic science is totally different from the normal science. That's why we are having two physics. Okay, that is normal physics, what you study, that is for bulk and other stuff and micro level and all. But when it comes to the atomic scale, you have a different physics that is called as quantum physics or quantum mechanics. So same quantum mechanics applies at nano size particles and also our molecules and the atoms, whatever we use. That's the reason the nanotechnology plays a very important role. That's why we are having nano-sized reinforcement here, because if you want to play with molecules, you have to go lesser than scale of that is nanoscale. So along with the nanotechnology, we can tune the property of the molecules. We can make hydrophobic property into hydrophilic property, hydrophilic property into hydrophobic property. These all things is possible by the nanotechnology. Nanotechnology plays a very important role and it has been widely used in every research. Similarly, same applications in order to enhance the properties of biopolymers, we can use the nanotechnology or nano-sized reinforcements. They will enhance the properties and the practical applications of the biopolymers. So biopolymers are being researched for its use in more and more ways possible. That means in the research and development, these all things are going on in the background. So examples of biopolymers are, we already have known in the previous classes, that is protein, starch, cellulose, DNA, RNA, lipids, collagen, carbohydrates, etc. Fibrin, actin, all those things. Types of biopolymers, we are having based on different things, we are having different types of biopolymers are there. So biopolymers can be classified into according to various scales. Some of the classifications are based on their type, their origin, and number of monomeric units and degradability. Based on their type, we are having sugar-based, starch-based, cellulose-based, synthetic material-based, because different types of the molecule are different polymers. And on the basis of the origin, we are having natural biopolymers, synthetic biopolymers, and microbial polymers. And having natural polymers, these are all natural biopolymers biosynthesized by living organisms directly. Whereas synthetic means these are polymers which are made up of renewable materials like polylactic acid, which are degradable in nature. It should be degradable. So not degradable, it will not come under this particular thing. So microbial biopolymers produced by the microorganisms. So on the basis of the monomeric units, we are having polysaccharides, proteins, and polynucleotides. Whereas polysaccharides, we know that the carbohydrates, the chain, which are branched or are linear, for example, starch, cellulose, and all. Next, proteins, we are having under polysaccharides, we know that mono, disaccharides, and all are there. Here it will be polysaccharides. Next, proteins, polymers which are made up of amino acids, and example, we are having the collagen, fibrin, etc. Right? So next we are having the proteins, done. So polynucleotides like nucleic acids, they are all long polymer chains composed of 13 or more monomeric units, example DNA, RNA, and etc. See, on the basis of monomeric units, how we are going to explain the polymers. So differences, this table is again studied in the previous class, you can just go through. Whereas biopolymers are found naturally, here it is man-made, generally degradable, it is not degradable, monogenicity. Here the polydispersity, complex structures, generally simple structures, renewable sources, here non-renewable sources, non-toxic and toxic. Next topic we are having the applications of the biopolymers that we will discuss in the next class.